What I thought I wanted to do was give a very broad introduction to the topic of natural selection and adaptation. So I'll talk about some of the common methods people are using to detect natural selection, and also a bit of about the models that people are doing or using to detect natural selection and model natural selection in general. And the target audience for this is really people that come from the mathematical sciences that have very little or no background in population genetics, and give you a flavor for how population geneticists are thinking about these different problems. And I would say, since I cover many different topics, I would more sort of give you some things that you could go and learn about later and look up, rather than actually really going in depth very much with each topic. So hopefully you'll be able to live with that. But it's a very broad topic, and it's certainly something that you could teach several semesters about just this topic. And there's thousands and thousands of papers on natural selection. You already heard a lot about natural selection. I know when Steve Evans was covering diffusion models, he was talking about natural selection. Here for the speciation discussion by Chuck Langley, he was also talking a lot about various forms of natural selection. So you already heard some about that. And hopefully this will help sort of put things together in terms of how do population geneticists think about this. So I'm going to start at the most basic level, what you would have learned if you're taking an introductory course in evolution. What would you have learned about models of natural selection there? So the very simplest model we can use, now I can see my slides are getting screwed up here. Well, that we could use, maybe we'll go to the Diploid model then, since the slide is not screwed up there. The almost simplest model we could use would be a simple Diploid model where we have two alleles in it, so there are three possible genotypes: capital A, lowercase a, and lowercase a. If the frequency of the capital A allele is p, then by Hardy-Weinberg equilibrium, we can, if there's random mating, predict the frequencies of these three genotypes in the population. What is different from natural selection from other models is we break with the idea about exchangeability. It's not so that the expected number of offspring from each individual is independent of the genotype, but that there's some fitnesses where the expected number of offspring in the next generation depends on the genotype of the individual. So when we talk about natural selection, very sort of broadly verbal definition would be that individuals, the expected number of offspring in the next generation depends on your genotype. Okay, so the fact that some individuals have many offspring, others have few offspring in the next generation, that's not natural selection in itself, except if it's because of some heritable differences between these individuals. Okay, so when we assign fitnesses, we often like to do it in terms of a selection coefficient, and one way it's done sometimes in the Diploid model is like this. You standardize with respect to one individual, say that individual has fitness 1, and then or one genotype, so the capital A, for example, in this case, has fitness 1. The heterozygous has fitness 1 plus HS, where S then is a selection coefficient, and H is a dominance factor, and the lowercase a genotype has fitness 1 plus S. So when people talk about selection coefficients, they come from this. This is one parameterization. There are different other ways of doing it as well. We can then, so what are these fitnesses? Right now, you can think of them as just simply viability, the chance of surviving to the age of reproduction. Okay, so we can calculate then the average fitness of in the population by taking the frequency of each genotype and multiplying by the fitness of that genotype, and then summing over all genotypes. And from that, we can then easily calculate the expected allele frequency in the next generation by taking the frequency of the capital A individuals, multiply them with their fitness, and half the frequency of the heterozygotes, and multiply with their fitness, and standardize then with the average fitness in the population. That will give us the allele frequency in the next generation. Okay, so that's sort of the simple Diploid model that you might start hearing about if you were taking an introductory course in evolution or maybe in population genetics. Okay, so I'm going to introduce some work that evolutionary biologists like to use when they talk about models like this. So for example, when they talk about directional selection, that means that one of the alleles are always favored, so that the fitness of one homozygous genotype is larger or equal to the fitness of the heterozygous type. That's larger or equal to the fitness of the other homozygous type. Okay, and a special case is neutrality, where all the fitnesses are the same. We don't usually include that in the condition for directional selection. So this is called directional selection because it favors uniquely one allele. And of course, what will happen in systems like this is that in the deterministic system, that the capital A allele will go to fixation eventually. So it'll always increase in frequency. Then you have this situation that's called overdominance, where the heterozygous have a fitness that's higher than either the homozygous type. And of course, in a system like that, it's very easy to convince yourself and that there will be a stable equilibrium, and the equilibrium point will then depend on exactly what these fitnesses are. And then we have a case that's not perhaps seen very often in nature, which is underdominance. That's where both of the homozygous types fitness are larger than the heterozygous type. And so then there will be an unstable equilibrium point, and either the capital A or the lowercase a will be fixed in this deterministic system. So one way to look at it is like this. If you have the allele frequency here, P, there will be some point to the right of that, the allele frequency will increase. To the left of that, the allele frequency will decrease. You can also take the average fitness in the population and plot that. That's what you have in the blue curve. And this illustrates something of importance, which is that natural selection will tend to increase the average fitness of the population, which is one component of what's known as Fisher's fundamental theorem on natural selection, which says that the increase in the change in allele frequency is given by the variance, additive genetic variance for fitness in the population. And part of that is then that change in allele frequency will always tend to increase fitness in the population. All right, so these are sort of the very simplest models that population geneticists would use and introduce in an undergraduate class. And then we'll talk about many other types of selection. Okay, so I'll just run through some of the concepts that the biology use when they talk about different forms of selection. So far the selection I was talking about was viability selection, so it operates on survivorship. But you could also have fertility selection that would operate on the level of finding mates, and fitness operates on the pairs that you made up with to produce offspring in the next generation. And there are lots of different models of fertility selection. Okay, related to that is sexual selection, which is selection related to actually finding mates, so that the chance of finding a mate depends on your genotype. Then there will be sexual selection. And the idea of sexual selection goes all the way back to Darwin, who used the idea of sexual selection to explain some of the extreme traits you see typically in males. For example, the long feather of the peacock. Why would a peacock have this enormously long tail? It can barely fly. It's subject to predation. Well, the verbal argument that Darwin gave was that, well, if first a female tends to prefer males with long tails, then males that have long tails in average will produce more offspring, and therefore natural or sexual selection will favor males with long tails. Okay, so Fisher then formalized this and showed that you can get this runaway process where there will be this reinforced effect where you will keep selecting more and more for longer and longer tails of the peacock until at one point the fitness disadvantage of having the long tail that you're subject to so much more predation will work against it, and the process will stop. But that is Fisher's runaway model of sexual selection. So in real life, in many of the traits we're interested in, sexual selection is really important, but I'm not going to talk more about it here. Then we also have various other models of, for example, frequency-dependent selection and fluctuating selection that differs from the model I talked about before by allowing fitnesses to change through time. In frequency-dependent selection, your fitness depends on the allele frequency. Okay, so that's a type of model that's relevant, for example, for interactions between parasites or pathogens and their hosts. Okay, where if you're a common parasite or common pathogen, the population might gain immunity to you, and so that type will be selected against, and there will be less immunity against the rare types. So in that way, selection will depend on the allele frequency. Fluctuating selection includes various different models that allow for various reasons selection or the way selection operates to change through time. So fitnesses get reassigned or somehow changes through time. And there's a lot of interesting dynamics with that. Something that I think people working on the mathematical side of population genetics were very interested in, some years ago, but has not lately had so much focus as it used to have. And I think there's a lot of interesting things perhaps to do with that for people that are interested in more mathematical problems. Then something that's very, very important for understanding social interactions and social behavior is kin selection. So the idea is that an allele of a certain type might give you a fitness advantage, not by improving fertility or survivorship of yourself, but of individuals that are related to you. So if there's a mutation arising in me that will give increased fitness advantage to all of my siblings, but not to me, that mutation will have a large chance of spreading in the population because that will, in the future generation, tend to be shared between my siblings, for example. So that fitness can operate not just on the individuals, but also on individuals that are related to me or to another individual. Okay, so that's kin selection, and of course it's important for is understanding things like apparent altruistic behavior, animals that live in social groups, why are some willing to sacrifice themselves for the social group they live in. That's typically most commonly explained because it has related individuals in the group. That it gives it sacrifices some of its own fitness for those individuals. And of course, it's an extreme case of that is social insects, where some individuals that forgo reproduction altogether, so they don't reproduce at all. And so they have, in some sense, they don't leave any offspring next generation, but because the queen will leave more offspring next generation, traits that favors that kind of behavior are still advantageous. We have also other types of selection. Group selection, for example, that's models in which you selection acts on the level of the group. So you have a group of individuals, different groups of individuals, and they compete with each other. Those models are usually a little harder to make work, but there's another set of models that people are working on. Very rarely in nature, I think, do you see group selection without a certain degree of kin selection. So I think most people these days, not really an area I work in, but I think most people these days think that kin selection is very common, but group selection without kin selection is probably very rare. And there are many more types of selection. Chuck was talking about selection at different levels, where you could have segregation disorders. It's another form of selection that can spread through the population, so you get genomic conflicts and so on. And so we have a very long list of different ways of different types of selection, different ways selection can operate. All right, but after that, I'm just going to go back to our very simple models. Hopefully, convince you that we have, a rich and complicated world of both. Now, what I give you verbal models that also can be formalized mathematically. But most of the time, when population geneticists work, and most of the work they're doing, they think about very simple models, such as this one here. And I know Steve Evans talked a lot about simple models like that, how they can be incorporated into, for example, the diffusion theory that we often use to understand population genetics. So to make things very simple, we can assume a multiplicative effect of fitness, genic selection, in which the fitnesses of the three genotypes are 1, 1 minus s, and 1 minus s squared. So they're very close to 1 minus 2s. Okay, in that case, the diploid model reduces to a haploid model. Okay, so now it's very easy to incorporate selection in the models you've heard about. In the Wright-Fisher model, for example, instead of sampling parents in the previous generation with equal probability, you sample them according to their fitnesses, proportional to their fitnesses. And we can put that into the equations we had before. We'll get a similar expression that the allele frequency in the next generation is given by this. This is the selection coefficient defined like this, and the allele frequency in the previous generation. And then we can write that as x plus x times 1 minus x times s plus something that goes to 0 when divided by s, and s goes to 0. So these are terms that would vanish if s is small. We can then I'm just reminding you, reviewing some of the things that Steve talked about. We can then get some, we can then derive some diffusion processes. So we can look at the expected change in allele frequency between 2 generations. If we now scale time in terms of n generations, then it'll be a time step of size 1 divided by n, which is equal to, from the previous slide, x times 1 minus x times s, and we multiply and divide by 2n. So gamma here is 2ns. And also we'll have a variance term that comes from the binomial sampling, similarly. And from that we can then define derive this diffusion process, where we'll have a term that has to do with the variance, and a term that's to do with the bias in the process, the selection here. And from that, what Steve was telling you about was that there are a lot of things we could do. We can look at, if we introduce a new allele with a certain selection coefficient acting on it, we can calculate what's the expected time spent at a certain frequency, what's the expected time until fixation, the expected time until loss, and so on. And probably most useful in many cases, the probability that will go to fixation. We can calculate that under simple models like that, and get very simple approximate expressions for that. And that's being used for many, many different things that we're doing in population genetics. Okay, so that was sort of the basic, kind of very simple model that population geneticists use, where you have, in this case, 2 alleles. You can generalize it also to k alleles if you want to, and so on. But these 2 allele models are being used a lot. And the reason why they're good for analyzing population genetic data, DNA sequence data, is often we're simply interested in a new allele that's being introduced in the population, and then the trajectory of that allele. And for that, all we need is really a 2 allele model to be able to understand that. So slowly moving towards looking at some of the models that people more use for analyzing data. So the one model that has become very popular for analyzing DNA sequence data is a set of models called Poisson random field models, introduced first by Sawyer and Hartle in '92. And what they want to do is they want to look at a process in which new mutations arise at a constant rate in time. So you have a Poisson process at which new mutations arise. And then they follow the path of the mutations, then in time follow this simple diffusion model. And then you integrate over all paths and over all times and ask, okay, what's the probability of seeing a mutation with a certain frequency in a sample? Okay, so the way that is done is that you have some density of allele frequencies, and it is it's not a stationary distribution of allele frequencies. We are operating here on an infinite sites model, so mutations, they are introduced and now go to fixation or loss, but they don't. There's no there's absorbing states. There's no stationary distribution here as such. But we can nonetheless, by looking at the time interval in which you are, or the amount of time you are spent at a particular frequency on your path to fixation or loss, we can get the expected proportion of alleles or allele frequencies in a certain category.
Just a clarification, if you exclude 0 and 1, then in station G you're looking for.
Well,
yeah, G doesn't integrate to 1, G integrates to infinity.
So it's asymptotic.
Well, it's not a stationary distribution, right?
I mean, it's in the sense it's a Markov process with absorbing states. In the same sense, it's not an ergodic Markov chain in that sense. I mean, that's all I'm saying there. But of course, you can find it. What does have a distribution you can get is the chance that some probability on a random sample of a mutation conditional on its segregating, for example, if that's what you are after.
No, just that you are still looking for a steady state. Are you looking for a steady state?
Okay, yeah, and that's it. I'm looking at a steady state. Like I'm looking at infinite amount of time. You introduce new mutations. The path of each mutation is described by a Markov chain that with 2 absorbing states, right? And then I look at a particular point in time, what's the distribution of allele frequencies of mutation at that point in time, and then integrate over all times. That's one way to think about it.
I mean, the Markov chain doesn't converge to equilibrium. It goes to either 0 or 1.
No. So there's 2 important, several important time scales. In the absence of the S, there are 2 important time scales. One of them is over n, and one is order 1 over mu.
Mm-hmm.
If you go for times much bigger than n, then you'll get this as a transient steady state, right? But much more than 1 over mu. So if you think, I mean,
well, I'm not sure we disagree. I mean, my statement about it not being ergodic is just the path of each single mutation, right? But I mean, you can define other functions of that process that has some kind of steady state distribution, clearly. All right. Anyway, so you can derive a density of mutations, and then you assume you assume a two-allele model, you sample binomial from that, and then you integrate over the binomial distribution over that density, and that will give you a sample distribution of what's the probability of seeing the derived form, the mutant form, seeing a certain number at a certain frequency. Okay, we can get that. So now we can do things like this. We can calculate what's the expected distribution of allele frequencies under different models, under different assumptions. So, for example, the classical, if you have a standard neutral model, so under the standard diffusion process without selection and infinite size mutation, you'll get a distribution that's proportional to 1 over x. Now, if you add selection to that, you'll get some different distributions. The distribution of allele frequency will be skewed to the right if you have positive selection, and to the left if you have negative selection. So I should say these distributions are standardized so that they integrate to 1 for all of them. Of course, there will be, if there's a lot of selection, you will actually tend to have, for the same mutation rate, fewer mutations segregating at any point in time. All right, just to make sure that everybody understands what we're looking at here, it's a sampling distribution where you imagine you're taking sample 20 chromosomes, so if you're diploid, 10 copies of the DNA, and then you look at what's the frequency in the sample of mutations of, what's the proportion of mutations in the sample where the derived allele segregated is a frequency of 1 out of 20, 2 out of 20, 3 out of 20, and so on. Okay, so we can derive these distributions. One important thing to know with this is, of course, beside all the simplifying assumptions that we have made right now of a constant population size and random mating and so on, we're also making one additional assumption here. Is that there's no interaction between different mutations. Okay, so we just get the marginal distribution for each mutation. In real life, there might be interaction between the mutations because they might be sitting on the same chromosomes. That's something that's being ignored. But you can fit this to data. So for example, this is from some data we analyzed a few years ago of 200 exomes. So exome, that's a part of the genome that codes for a protein. So you can it's easy and cheap now to pull out all those parts of the genome and sequence that for a large number of individuals. And then you can look at the distribution of allele frequency you see there. So this is for humans, for some Europeans. And what you have in the blue here is the neutral expectation, the ones that's proportional to 1 over x from before. And then you have in the real data 2 different distributions. The green one is synonymous mutations, and the purple one, that's non-synonymous mutations. And just let me remind you what's the difference between synonymous and non-synonymous. Remember that the central dogma of molecular biology is that you have DNA that's been transcribed to RNA that's been translated into protein. A triplet of free nucleotide codes for 1 amino acid in the protein. Okay, so you have 64 possible triplets, but there are only 20 amino acids. So there's some redundancy. So you can have mutations, 2 mutations that occur in a coding region, but the resulting protein is not changed. Okay, so those are the synonymous mutations. The ones where you have a mutation that actually change the protein, those are the non-synonymous mutation. We think mostly selection acts at the level of the protein. The proteins are the ones that are doing the work in the cell. Okay, so most selection should work at that level. So we often use the synonymous mutation as a neutral standard, as what you would expect if there was no natural selection. And we see in this particular case that for these data that the synonymous and the neutral one are maybe not so far away from each other, but the non-synonymous ones, they have this strong excess of rare alleles, and that's exactly what you would expect if many of them are under negative selection. And you can then take these Poisson random field models, which are very simple, and you can make some even more assumptions about the distributional form of mutations, and then get some kind of estimate of how much selection would you need to be able to get such a difference between synonymous and non-synonymous mutations. Okay, so this is, for these type of data, you would find that about 80% of all non-synonymous mutations need to be under some kind of selection to be able to explain the data. So that's a lot. So many mutations segregating in the human populations that is under selection right now. Okay, so this is all what I talked about so far. So this is on an assumption of a gamma distribution of selection coefficient plus a point mass at 0. So you just let there be a point, something that are strictly neutral. So you have point mass 0, and then an additional gamma distributed number of mutations that are of minus s of selection coefficients. And then you ask how big a proportion of the probability mass go into the gamma distribution, how much go into the point mass at 0. And you can use all distributional form and you will get different answers. But I mean, so I don't think I would, whether it's 80% or 75% or 85%, I don't think that will depend on the distributional forms. But
there is there's a general thing about the distribution, right? Is that unless you have very large samples, this is entirely sensitive to ones where the S is of order 1 over n. Right?
So you can incorporate also the ones that don't go to fixation, right? So the fact you also have information about the invariable ones. So the ones that you see segregating, so the ones that you actually see segregating in the population are the ones where it's on the order of 1 over n, right? But there's also information here about mutations that you never see segregating in the population. That information we also have there, having this. Most mutations that are under selection, or many positions in which the mutation that would be under selection probably would never see anything segregating there because the effect of that mutation is so deleterious. So that's incorporated here as well in that estimate. All right. So what I've been talking about so far are what I would call the direct effects of natural selection on a mutation. So we're modeling a particular mutation that's under selection. Okay, but most of the pattern you would see in real data is actually of what you would call the indirect effects of mutation. That is the effects of a selected mutations on nearby neutral variants, on positions in the genome on which there's no selection acting. And much work in population genetics these days, in the past maybe 15, 20 years, have really focused on understanding that pattern. Not so much on the selection, on the mutation themselves, but the effect of the selected mutations on other variants that are not under selection. Okay, so if you have an advantageous mutation that occurs in the population and increases in frequency, and it goes to fixation in the end, to talk about a selective sweep. And just sort of a little intuitive way of understanding what's going on. So you imagine here you have some DNA sequences that are segregating in the population. Every position where you have a star means that there's a mutation there. So this is the derived allele in that particular position. You imagine that at some point in time, there's a new advantageous mutation that lands on a chromosome. If it's advantageous, with some probability, eventually it will go to fixation, so all individuals in the population will carry that mutation. But if there was no recombination, then it's not just that mutation that goes to fixation, it's all the linked mutations, these mutations that were sitting on the same chromosome as the advantageous mutation. Okay, so that's the pattern that we're looking at. We'll get this pattern where the effect of the advantageous mutation is not just on itself, on the selection on that, but it's also on the nearby sites. All right, and I'm just going to remind you about the process of recombination. I know other people talk about recombination, but it's really fundamental for understanding anything in genetics, recombination. So you have, during meiosis, you have the paternal and maternal copies of the DNA, and they can swap DNA between them, the chromosomes that you inherited from your mother and your father. So when you produce gametes, egg cells, sperm cells, part of the DNA you pass on to the next generation for a particular chromosome. Part of it might be from your mother and part of it from your father. Okay, so that's the process of recombination, which is, I know I could see from slides that you have heard about before, but I just want to remind you. So because of that, you can have what people call escape by recombination. You can imagine there was a recombination event between, say, this chromosome here, and this mutation you have over here is being put on this chromosome. So after this advantageous mutation has gone to fixation, in this position, you still have the two alleles, the derived and the ancestral ones, because of this recombination event that occurred as the advantageous mutation increased in frequency in the population. Okay, so the pattern, so we'll go a little bit into what are the models that people use to model that, but I'm just first going to explain a little bit about the pattern that people expect to see because of this, which is mainly a reduction in variability in the region around the advantageous mutation. And why is that? That's because right around the advantageous mutation, all mutations have either been lost or gone to fixation together with advantageous mutation. So if you look at the amount of variability in the genome, and there's been a particular position in the genome where there's been a selective sweep like this, you will see this a reduction in variability like that. And it will also affect other patterns in the data that I'll talk about a little bit later. Okay, so this idea go back all the way to a paper by Main, Smith, and Haig from 1974. But the modern way of thinking about this problem and modeling it can be, I think, traced back to this paper by Kaplan, Hudson, and Langley from 1989. So they were interested in and made one of the first models of coalescence models with a selection. And the way they were modeling it is that they were dividing the problem, and that's something that's been used in many, many applications since then, dividing the problem into two parts. The first problem is what's the trajectory of the selected allele through time? How does that change? So that's what you have here depicted here. And the second problem is then how does the genealogy then look conditional on that trajectory? Doing it like that simplifies things a lot. So what you have here depicted in this figure is a coalescence tree for a linked neutral site. And the way we interpret it is that if there was no recombination, of course, all of the lineages would have to coalesce at the time, at or before, looking backwards in time, the advantageous mutation arose. At that point, there was only one copy of the DNA. So if there's no recombination, all lineages must coalesce back there. But if there's recombination, you can escape the selective sweep. And you might have a coalescence times that's older than that. Okay, so what Kaplan, Hudson, and Langley did was they divided the process of selective sweep into three stages. A stage first in which the allele frequency is very low, then an intermediate stage here in which the allele frequency increase, and then a last stage in which the allele frequency is very high. And the reason why they did that was that they were saying, okay, this part of the trajectory is almost deterministic. So let's model that using a deterministic model, and just then deal with the stochasticity down here and up there. Okay, so we have the three phases. First, a phase at which the allele is of low frequency, where the allele frequency is then modeled as a supercritical branching process. So there's a positive selection, so it'll be a supercritical process. Okay, and the idea then is, an idea that people subsequently has used a lot, is that that when the allele frequency is very, very low, you can model the distribution of allele frequencies using essentially assuming independence. So you can model it using birth-death processes, or the tree as using a simple branching process. As the allele frequency become larger, that doesn't work anymore. But for lower allele frequencies, that works. Then you have the deterministic change. That's the same equation, slightly different form as we've seen before now, as a deterministic change through time. And then you model the 1 minus the allele frequency as a subcritical branching process as you get to very high allele frequencies. Okay, so you have those three phases. Now, what do you do then conditional on the coalescence process, conditional on the allele frequency? Okay, so then you use, and that's another thing that people have been using a lot subsequently, is that you use a structured coalescence then. And I know you already heard about structured coalescence processes. So those are the processes you usually would use when you have two or more demes or subpopulations. Here you can think of the subpopulations as being the different allele classes. The neutral allele linked to the selected allele and the neutral allele not linked to the selected allele. And then you can model, conditional on that allele frequency trajectory, you can model the coalescence process as a time inhomogeneous Markov jump process with the following rates. So you'll have coalescences in one allele class at the usual rate. If there are I individual, I choose 2, but then divided, standardized not by only by the population size you need to standardize with is the allele frequency times the population size in that allele class. The other allele class the same. You have coalescences occur at the same rate as you normally would, but you need to standardize by the allele frequency of that allele class. And then you also have what takes the role of migration events between the two populations. And that's recombination events that occur at rates that are proportional to the number of alleles in that allele class. And then also proportional to a recombination rate. So this is exactly like a structured coalescence model. You just, instead of having migration, you have recombination here. All right. So coalescence trees, how will they look? Well, if this is a normal neutral coalescence tree that look like that, on a selective sweep, what this type of model would predict is that you'll have something like this. You'll have a short selective phase as an advantageous allele increase in frequency. Many of the lineages are forced to coalesce during that time period, but then by recombination, some can escape. So you'll get trees that may look something like this, coalescence trees. And then also, you can now distribute mutations on those trees and then predict what the distribution of allele frequencies then would be, and you'll get something like what you see in the yellow columns. If you take a window around the selective sweep, you'll find that there are increase in mutations of low frequency and of high frequency. These are the ones that hitchhike with advantageous mutation. These are the ones that where selection that initially were not linked with advantageous mutation, so they decreased in frequency. Okay, so you get sort of this bimodal distribution of alleles. All right, so there's been lots of work on these models. So Wolfgang Stephan, for example, has done lots of work on these models, and one of the things he was doing in some of this model was saying, okay, the stochastic phase doesn't really matter much because the allele frequency there is so low, there's not much opportunity for recombination anyway, so we can kind of ignore that. So let's focus on the deterministic model. And then if you do that, things become easier. You can derive a lot of interesting results. Other people have been looking at what's happening more during the selective phase, and what's the chance, can you get a probability distribution of the number of lineages that are actually coalescing during the selective phase. And there's, for example, a paper by Rick Durrett and Swansberg in 2004, where they show that there's a limit in which the number of lineages that are forced to coalesce is simply binomial. And so you can, from that, it becomes very, very easy to do various kind of calculations if you work in that limit, because you have certain lineages that will look like as if there hadn't been selection, and there are certain lineages that are all forced to coalesce with this binomial probability. Of course, this is an approximation that ignores the fact that, in fact, all the lineages don't coalesce at the same time, but they will coalesce at different times in different clusters, and there are lots of work also on that, on sort of the theory of coalescence process with multiple mergers in the context of selective sweeps, and these are some of the references to some of that work. Okay, all right, so this is, so I just want to talk a little bit about generally about the coalescence approaches to selection. So what we saw here was that what was done was that you conditioned on the allele frequency and the trajectory of allele frequencies, and then you would have a coalescence process that runs like a normal coalescence process within each allele class. Okay, and then you could, by recombination, go between these allele classes. That kind of way of using a structured coalescence, conditioning on allele frequency, has been used many, many times. It's a common way of dealing with natural selection that's convenient when there are fewer allele classes. And for example, for certain type of very strong balancing selection is selection that maintains variability in the population, where you don't predict that there's much of a change of allele frequency through time. These type of approaches are particularly appealing and lead to simple results. Then there's another type of approach, which is what people use, for example, linear birth-death processes for rare alleles. So as I talked about before, you can model the change in allele frequency, or equivalently the coalescence process, using models that assume essentially independence among the lineages, as long as the allele frequency is very low. That works. So there are a number of papers on that. Finally, there's a set of approaches which use what's called the ancestral selection graph. And I'll just end up this to talk a little bit about ancestral selection graph. Okay, so this is a sort of a full coalescence model for selection, I would say. Maybe a little bit more principled approach, perhaps. Why is it hard to do the coalescence with selection? Well, it's hard because, as we've seen, that the rate of coalescence depends on which allele class that you're in. and you need to know the allele class, and that it furthermore depends on what's the population frequency of that allele class, which usually will be unknown, okay, and can change through time. So the way in the ancestor selection graph to deal with that is that you're gonna stick in some extra fake branches in the coalescence tree. These branches are gonna essentially mimic the effect of selection. So if you know that you have 5 capital A alleles and 5 lowercase a alleles, and know that capital A alleles are strongly favored, then you would expect, looking backwards in time, coalescences occurring at a higher rate among the capital A alleles. Okay. And looking forward in time, if you look at the reverse process starting at an ancestor of a tree, it might be more intuitive to realize that if you have 2 lineages, one that carries the favored allele and one that carries the unfavored allele, you would expect more populations or more splitting of lineages, more branching of lineages for the favored allele than for the unfavored allele. Okay, so you can deal with that problem rigorously by putting in fake branches in the coalescence tree, where you'll have to make a choice between which branch you actually should choose depending on which allele is assigned to that branch. So you'll simulate, if you want to simulate a process like this, the way you would do it is you would simulate coalescence events like normally. So they occur at rate j times j minus 1 over 2, or j choose 2 if they're j lineages. But then the new thing is you have branching events that occur at a rate sigma j divided by 2. This is this selection parameter 2ns, the same thing as gamma from before in the very simplest models. And then, so these, if you look at this tree, or this genealogy here, you have these branching event here, and then you label the 2 new branches, one as being the incoming branch, and then and another one as being the contributing branch. What is it called? The incoming branch and the contributing branch, I think. Continuing branch, thanks. Continuing branch. Okay, and so the next thing you want to do, you want to then put mutations on the tree, including both the continuing and the incoming branch. And then what you do is that you first start at the most recent common ancestor of the entire sample, so at the root. And then you choose the allelic state there according to the stationary distribution. Okay, so you assign a allelic state there. And then you put mutations on the tree, as you usually do, using, if you are in the infinite science model, a Poisson process or some other mutational process, along the, and superimpose that on the edges of the coalescence graph. And then what you want to do is, after you assign that, you want to choose whether you should erase an incoming or continuing branch. An incoming or continuing branch. And that will depend on the allelic state that then has been assigned to those. So if there's a conflict between these 2 branches, so they have different allelic states, you always choose the allelic states of the favored allele. Okay, so you can show that that correctly generates the coalescence tree with selection. Now, a couple of, so you, in some sense you have a fantastic framework here, a rigorous framework for describing the coalescence process with selection. Why hasn't it been used so much? Well, one of the main reasons is that computationally, it can only deal with quite weak selection, at least in the way it's formulated here. So you have coalescences that occur at a rate that's about quadratic, and you have branchings that events that occur at linearly that's linearly, the rate of that is linear in the number of lineages. So that will guarantee that eventually you'll get to a common ancestor for all lineages of down to 1 lineage. But if this coefficient here is high, it can take a very, very, very long time. As soon as you're down to very few lineages, you still have could have a high rate of branching. So you will have to wait a very, very long time until you're back to just having 1 lineage. And in practice, if sigma here gets to be 10 or larger, this starts to become very computationally bothersome. And so that's one of the main reasons why this framework, despite its, as appealing as it is mathematically, perhaps has not been used very often for actually making inferences in population genetics.
Is this object the same as the ancestral recombination graph?
No,
But you're going left and right,
no,
depending on the...
no. This is completely different. In
But I
the
mean, it's a branching process, and the rate of branching is the same if you have rho instead of sigma. But that's
yeah, yeah. I mean, there's similarities in that sense. Yeah, but I mean, it's fundamentally different. This that you actually don't know. So this where you delete either the incoming or the continuing graph. That will actually give you a coalescence tree for marginally for one particular
if you pick a
position.
sequence,
So,
then
but
the R gives you a tree.
yeah. So
But this is just about one position.
this is just a one position, and so yeah. So you're looking at one particular position, and this is not a graph that represents the ancestry. This would be one that represents the ancestry. You have to delete these edges of the graph first.
Yeah, the extra non-tree edges, right, are just giving you something that is
it's like a
Yeah.
keeping device that produces the selection. and
Yeah,
don't
yeah.
have any sort of meaning in terms of genealogies.
Yeah. All right. So I think perhaps if you're a mathematician, I think these type of models and seeing are there ways how can we better deal with these kind of problems where the size of the graph doesn't explode as we look at stronger selection.
So, comparing this to the structured coalescence approach, so there you are actually dealing actively with linkage.
Yes.
So here, is there again a way of trying to add linkage onto this?
Well, in the structured coalescence approach, yes. Well, usually people have not been doing that, but you can do that because you can simulate the marginal tree for one side that's selected, and then conditional on that simulate for the linked trees with a recombination graph. And that has been done a little bit, but not much because it's kind of complicated and cumbersome to do. The structured coalescence is also used not just for linkage. I mean, it is used for just also looking marginally at one side. So in which case the rate of you can put instead of recombination mutation, where you can take one from the other. Instead of it's a recombination, it's a mutation event that take you from one class to another class. So for balancing selection, that will be the usual interpretation. All right, so I think we should take a break now if there are no more questions.